April 25, 1953. The journal Nature publishes 3 papers on the structure of DNA. 3 separate articles printed one after another. The first paper, the one everyone remembers, is by James Watson and Francis Crick. It describes the double helix structure of DNA, the most important biological discovery of the 20th century. The second paper is by Maurice Wilkins, Supporting Data. The third paper is by Rosalind Franklin and her graduate student Raymond Gosling. More supporting data. 3 papers, equal billing. Except that's not what happened. Watson and Crick got the credit. Their names became legendary. Nobel Prize winners, giants of science. Wilkins got a share. He was there. He helped. And Rosalind Franklin, she became a footnote, an assistant. The woman who took some helpful pictures, but here's what actually happened. Rosalind Franklin took the photograph that proved DNA's structure. Photo 51, the most important X-ray crystallography image in history. She didn't just take it. She spent months perfecting the technique, calculating the angles, interpreting the data. She knew what it meant. She was writing a paper about it. And then Maurice Wilkins took her photograph without permission and showed it to James Watson. Watson saw it, understood it immediately, and he and Crick published first, using her data, without her knowledge, without her consent. When Watson finally wrote about it decades later, he didn't apologize. Instead, he described Rosalind as difficult, unattractive, and bad at science. She didn't even understand what her own data meant, he claimed. This was a lie, but by the time Watson published his version of history, Rosalind Franklin had been dead for years. She died in 1958 at age 37, ovarian cancer, likely caused by the radiation exposure from the very X-ray work that revealed DNA's structure. 4 years after her death, in 1962, Watson, Crick, and Wilkins won the Nobel Prize. The Nobel isn't awarded posthumously, convenient rule, so Rosalind Franklin was erased. Quietly, efficiently, 2 men became legends, 1 woman became invisible. But to understand how this happened, we need to go back to the beginning, to London, 1920, to a girl who loved logic more than anything. She was born Rosalind Elsie Franklin on July 25, 1920, in Notting Hill, London. Her family was wealthy, educated, Jewish. Her father, Ellis Franklin, was a merchant banker, politically active. A believer in social justice and education. From the beginning, Rosalind was different. While other children played, she organized, she categorized, she solved puzzles with an intensity that unnerved adults. At 6 years old, she was doing arithmetic for fun. At 8, she announced she wanted to be a scientist. Her father's response was mixed. He believed in education, but he didn't believe women should work. Education was for becoming cultured, not employed. Rosalind didn't care what he believed. At age 11, she enrolled at St. Paul's Girls' School, one of the few schools in London that taught physics and chemistry to girls. She excelled at everything. But she loved science, the precision, the logic, the fact that nature didn't care about your gender or your charm. It only cared about evidence. By 15, she knew exactly what she wanted, to be a scientist, a real one, not a hobbyist, not a teacher, a researcher. Her aunt asked her what she wanted to do when she grew up. "I'm going to be a scientist," Rosalind said. "But you're a girl," her aunt replied. Rosalind looked at her, genuinely confused. What does that have to do with anything? In 1938, at age 18, she won a scholarship to Cambridge University to study chemistry at Newnham College, one of only 2 women's colleges at Cambridge. Cambridge in 1938 was not kind to women. Female students could attend lectures and take exams, but they couldn't receive degrees. Not officially. That rule wouldn't change until 1948. Women sat in the back of lecture halls, were ignored by professors, were openly mocked by male students. Rosalind didn't care. She wasn't there to be liked. She was there to learn, and she was brilliant. Top marks in physical chemistry, exceptional work in molecular structures. In 1941, she graduated. Officially, she received a certificate. Unofficially, she had earned a degree in chemistry with honors. Then came the war. World War II shut down most academic research. Rosalind took a job at the British Coal Utilisation Research Association, studying the molecular structure of coal. It sounds boring, it wasn't. Coal was essential to the war effort. Understanding its structure meant improving its efficiency. Rosalind's work helped classify coals and optimize their use. She published 5 papers. Serious, respected work. Her research became the basis of her doctoral thesis. In 1945, the war ended. Rosalind earned her PhD from Cambridge. Finally official, she was 25 years old, a doctor of chemistry, ready to do real research. She moved to Paris to work at the Laboratoire Central des Services Chimiques de l'État. There she learned X-ray crystallography under Jacques Mering, one of the best in the world. X-ray crystallography is a technique where you shoot X-rays through a crystallized substance. The X-rays scatter, creating a diffraction pattern. If you know how to read the pattern, you can determine the molecular structure. It requires precision, patience, mathematical skill, and ability to see 3-dimensional structures in 2-dimensional images. Rosalind was extraordinary at it. She spent 4 years in Paris, the happiest years of her life, she'd later say. She was respected, treated as an equal, published important papers. But in 1951, she was offered a position back in London. A 3-year research fellowship at King's College to work on DNA. DNA was the next frontier. Scientists knew it carried genetic information, but nobody knew how it worked. What was its structure? Rosalind accepted. She returned to London, ready to solve the puzzle. She had no idea what she was walking into. King's College, London, January 1951. Rosalind Franklin arrived ready to work. She'd been hired as a research associate with her own project, using X-ray crystallography to determine DNA's structure. She set up her lab, calibrated the equipment, started preparing DNA samples, and immediately there was a problem. Maurice Wilkins. Wilkins was already at King's College, also working on DNA. He'd been there during the war. He thought of DNA research as his project. When Rosalind arrived, he assumed she was there to assist him, to be his assistant. She wasn't. She'd been hired independently. She had her own funding, her own project, but nobody had clarified this. The department head, John Randall, had told Rosalind she'd be leading the DNA work, but he never told Wilkins. So from day one, there was confusion, resentment. Wilkins treated Rosalind like a technician. She treated him like an irrelevant colleague. The tension was immediate. But Rosalind didn't care about office politics. She cared about the science. DNA came in 2 forms, a dry A form and a wet B form. Previous researchers had struggled to get clear images of either. Rosalind developed a new technique. She controlled the humidity with precision, stabilizing the B form. She adjusted the X-ray exposure times. She refined the mathematical analysis. And in May 1952, after months of work, she took a photograph. Photo 51. It's a black and white X-ray diffraction image. To most people, it looks like a blurry X. Smudged spots arranged in a cross pattern, but to a trained crystallographer it was unmistakable. The X pattern meant a helix. The spacing of the spots revealed the dimensions. The symmetry indicated 2 strands, not 1. Photo 51 was the structure of DNA, right there, visible. Rosalind knew it. She started doing calculations, measuring angles. She was methodical. She wanted to be absolutely certain before publishing. She wrote in her notebook, "Evidence for a helical structure." She was preparing a paper, writing up the data, being careful. And then Maurice Wilkins made a decision that would change everything. January 30th, 1953. James Watson visited King's College. Watson was an American biologist working at Cambridge with Francis Crick. Together they were trying to build a model of DNA's structure. But they were struggling. They didn't have good data. They were guessing. Watson went to King's College to see if Wilkins had anything useful. Wilkins was frustrated with Rosalind, who he felt was difficult and uncooperative, frustrated with the lack of progress. So when Watson asked if there was any new data, Wilkins did something unforgivable. He showed Watson photo 51, without asking Rosalind, without telling her, without permission. Watson saw the photograph and immediately understood. The instant I saw the picture, my mouth fell open and my pulse began to race, Watson later wrote. It was a helix, obviously, undeniably. Watson rushed back to Cambridge, showed Crick. They started building models, but they needed more data, dimensions, measurements. And then they got lucky again. Max Perutz, a colleague at Cambridge, had been on a committee that reviewed King's College research. He had access to a progress report Rosalind had written, detailed measurements, calculations. Data. Perutz gave the report to Crick. Now Watson and Crick had everything. Rosalind's photograph, Rosalind's measurements, Rosalind's data. They worked furiously, building models, testing configurations. On March 7, 1953, they figured it out. The double helix, 2 strands, anti-parallel. Base pairs in the middle. It was perfect, elegant, obviously correct. And it was based entirely on Rosalind Franklin's data. Data she didn't know they had. Watson and Crick wrote their paper immediately. They submitted it to Nature on April 2, 1953. In the paper, they thanked Wilkins and Franklin for stimulating discussions and acknowledged they were stimulated by knowledge of the general nature of the unpublished experimental results. That was it. One sentence, buried in the text. No mention that Photo 51 was the key. No mention that they'd used her data without permission. Just vague acknowledgement. Nature published the paper on April 25, 1953. It was 1 page long. It included no photographs, just a simple diagram of the double helix and a sentence that would become famous. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Translation: we just figured out how life works. The paper was published alongside 2 others, one by Wilkins, one by Franklin and Gosling, but Watson and Crick's was first. It was the one that got attention. Within days, newspapers were calling it the greatest discovery in biology. James Watson and Francis Crick became famous. Their names were everywhere. Rosalind Franklin went back to work. She didn't know what had happened, didn't know Wilkins had shown her photograph without permission, didn't know Crick had seen her data. She thought Watson and Crick had simply beat her to publication, that they'd figured it out independently. It bothered her. She knew her data was better. But she was a good scientist. She accepted it. In July 1953, her fellowship at King's College ended. She left, gladly. The environment had been toxic. She moved to Birkbeck College and started working on the structure of viruses. Brilliant work, groundbreaking. She published 17 papers on viruses in 5 years. She was happy, productive. And then, in 1956, she started feeling sick. Abdominal pain, fatigue. She ignored it. She was busy. By early 1957, it was worse. She saw a doctor. Ovarian cancer. Advanced. She had surgery. The cancer came back. More surgery. It came back again. She kept working. Between surgeries, between treatments, she published papers from her hospital bed. On April 16, 1958, Rosalind Franklin died. She was 37 years old. The cause was almost certainly radiation exposure. She'd spent years working with X-ray equipment, unshielded. The same work that revealed DNA's structure likely killed her. Her obituary in the Times was brief. Dr. R.E. Franklin, research worker in biophysics. No mention of DNA, no mention of Photo 51. She was buried quietly. Her family mourned. Her colleagues moved on. And 4 years later, in 1962, the Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick and Maurice Wilkins for their discoveries concerning the molecular structure of nucleic acids. Rosalind Franklin was not mentioned in the Nobel announcement, not once. Let's talk about what Rosalind Franklin actually discovered. Photo 51 wasn't just a helpful clue, it was the proof. Before Photo 51, the structure of DNA was speculation. Multiple models existed: single helix, triple helix, straight chain. Photo 51 eliminated all other possibilities. It showed a double helix with specific dimensions. period. Watson and Crick didn't discover DNA's structure through brilliant reasoning. They discovered it by looking at Rosalind's data and building a model that matched. This isn't speculation. Watson admitted it. In his 1968 book, The Double Helix, Watson wrote openly about seeing Photo 51. He described it as the moment everything became clear. But instead of crediting Rosalind as the key contributor, he portrayed her as an obstacle. Difficult, defensive, too emotionally attached to her data to interpret it correctly. He called her Rosie, a nickname she hated and never used. He described her appearance critically. She was not unattractive and might have been quite stunning had she taken even a mild interest in clothes. He suggested she didn't understand her own data, that she was too cautious, too slow. This was character assassination, and she couldn't defend herself. She'd been dead for 10 years. The book became a bestseller. It shaped how the public understood the discovery of DNA. James Watson, brilliant, irreverent genius. Francis Crick, his clever partner. Rosalind Franklin, the uptight woman who almost got in the way. But here's the truth, Rosalind knew exactly what her data showed. Her notebooks prove it. She'd written about helical structure. She'd calculated the dimensions. She was being careful, because that's what good scientists do. She wanted to be certain before publishing. Watson and Crick published fast because they had nothing to lose. They were building models based on other people's data. If they were wrong, they'd just build another model. Rosalind was generating the actual data. If she was wrong, it would destroy her credibility. So she was careful, methodical, rigorous, and she was punished for it. Because Watson and Crick published first, history gave them credit, even though their work was only possible because of Rosalind's photographs and measurements. The impact of DNA's structure on science is impossible to overstate. Understanding the double helix explained how genetic information is stored, how it's copied, how it's passed from parent to child. It launched the entire field of molecular biology. Within a decade, scientists had cracked the genetic code, figured out how DNA sequences correspond to proteins. By the 1970s, genetic engineering became possible. By the 1980s, DNA fingerprinting revolutionized forensics. By the 1990s, the Human Genome Project began mapping every gene in human DNA. Today we edit genes, we cure genetic diseases, we trace ancestry through DNA, we create mRNA vaccines. All of it traces back to that moment in 1953 when the structure of DNA was revealed. And that revelation came from Photo 51. The Nobel Prize in 1962 was worth approximately $50,000. Split 3 ways between Watson, Crick, and Wilkins. But the real value wasn't the money. It was the immortality. Nobel laureates become legends. Their names are remembered forever. They get statues. Buildings named after them. Their work is taught in every biology class on Earth. James Watson became the director of Cold Spring Harbor Laboratory, one of the most prestigious positions in biology. Francis Crick continued groundbreaking work in neuroscience. Maurice Wilkins had a long-respected career. All 3 lived into old age. Watson is still alive today, in his 90s. And Rosalind Franklin, she got a small plaque at King's College, installed in 1992. 34 years after her death. The Nobel Committee has a rule: prizes are not awarded posthumously. You have to be alive when the prize is announced. Rosalind died in 1958. The prize was awarded in 1962. Some have argued that if she'd lived, she would have shared the prize. Maybe. Maybe not. The Nobel can only be split 3 ways maximum. Watson, Crick, and Wilkins made 3. There was no room for Rosalind, even if she'd been alive. The committee could have chosen differently. They could have given it to Watson, Crick, and Franklin, leaving out Wilkins. But they didn't, and they never explained why. After Rosalind's death, her colleague Aaron Klug continued her work on viruses. He won the Nobel Prize in Chemistry in 1982. In his Nobel lecture, Klug spent significant time honoring Rosalind, explaining her contributions, making sure her name was said. It was the closest she ever came to being recognized at that level. In 2003, 50 years after the publication of the double helix structure, King's College finally acknowledged what happened. They held a symposium. They admitted that Rosalind Franklin's contribution had been minimized, that she deserved equal credit. Watson gave a speech. He admitted that he'd been unfair to Rosalind in his book, that she was a better scientist than he'd portrayed. It was something, a half apology. 50 years too late. But here's what Watson didn't apologize for: using her data without permission. He framed it as a misunderstanding, a lack of communication between labs. Just one of those things that happens in competitive science. But it wasn't a misunderstanding. It was theft. Maurice Wilkins showed Photo 51 to Watson without Rosalind's knowledge. That wasn't an accident. It was a choice. Max Perutz gave Rosalind's progress report to Crick without her permission. That was a choice. Watson and Crick used her data to build their model and published without telling her. That was a choice. These weren't mistakes. They were decisions made by men who didn't think they needed permission from a woman. And it worked. They became legends. She became a footnote. Here's what's most painful. Rosalind would have shared. Her colleagues at Birkbeck said she was generous with her data, collaborative, eager to work with others who respected her. If Watson and Crick had simply asked, she likely would have said yes. But they didn't ask. Because they didn't have to. Because she was a woman. Because they could get away with it. And they did. This still happens. In 2013, a study found that women scientists are significantly less likely to be credited as authors on papers, even when they do equal work. In 2017, the Nobel Prize in Physics was awarded for gravitational wave detection. Over 1,000 scientists worked on the project. The prize went to 3 men. In 2018, Donna Strickland won the Nobel Prize in Physics. She was only the 3rd woman ever to win it. When she won, she didn't even have a Wikipedia page. Male scientists with far fewer accomplishments had detailed biographies. In 2020, Andrea Ghez won the Nobel Prize in Physics for black hole research. Headline in one newspaper: "Mother of 2 wins Nobel Prize." When male scientists win, nobody mentions their children. The pattern continues. Women do the work. Men get the credit. Rosalind Franklin proved DNA's structure. Watson and Crick won the Nobel. Jocelyn Bell Burnell discovered pulsars. Her male supervisor won the Nobel. Lisa Meitner discovered nuclear fission. Otto Hahn won the Nobel. Chien-Shiung Wu proved the theory that won the Nobel. 2 male colleagues won. She didn't. Over and over and over. The Nobel Prize has been awarded to 621 men in science and 24 women. The problem isn't that women aren't doing the work. The problem is that they're doing it and being erased. Photo 51 hangs in museums now. It's one of the most famous scientific images in history. Rosalind Franklin's name is finally attached to it. Finally. You can buy posters of it. T-shirts, coffee mugs. Her face is on stamps. There's a European Mars rover named after her. Universities have endowed Rosalind Franklin fellowships, scholarships, awards. She's become a symbol. The brilliant woman who was robbed. And that's good. It's important that we remember her. But here's the question: does it matter? Does posthumous recognition undo the injustice? Does it give her back the career she deserved, the Nobel Prize she earned? Does it change the fact that she died at 37, never knowing that Watson and Crick used her data without permission? Does it change the fact that for decades she was barely a footnote? No, it doesn't change any of that. But it does something else. It shows the next generation of women scientists that they're not imagining it. That yes, this happened. Yes, it was wrong. Yes, she deserved better. And maybe it makes us more careful, more aware, more willing to ask, who did the work? Whose data was this? Who's being erased? Because it's still happening right now in labs around the world. Women doing the research, men taking the credit. And the only way it stops is if we refuse to forget. Rosalind Franklin took the photograph that revealed the structure of DNA. She did the calculations. She interpreted the data. James Watson and Francis Crick saw her work without permission and published first. They won the Nobel Prize. She got nothing. She died at 37. They lived to be legends. That's the story, the whole story. Not the mythology, not the everyone contributed equally version. The truth. And the truth is this: one woman did the work that changed biology forever. And two men took the credit. Remember her name. Not as a footnote, not as an assistant, not as the woman who took some helpful pictures, as Rosalind Franklin, scientist, crystallographer, the woman who showed us the structure of life itself. Photo 51, April 25, 1953. The most important photograph in the history of biology. Taken by a woman the world tried to forget. Remember her name.